let us learn about the structure and working of ear, the other sense organ of human beings, is otherwise called as statoacoustic organ. Why is this ear also called as statoacoustic organ? Because the ear, despite of performing the function of hearing or auditory function, also plays a key role in body balance. Here stato stands for staticity or body balance. Next, acousticity stands for hearing. Together, statoacoustic organ. Now, this ear has three parts. Ear of human being has three different parts. What are the three parts of the ear and what are the functions? The three parts of the ear are: number one, it is external ear. Number two, middle ear, external ear, middle ear and inner ear. Now, let us learn about what are different parts of each region. To begin with, external ear. External ear has three parts. What are they? Number one, it is ear pinna, otherwise called as auricle. Number two, it is external auditory meatus. External auditory meatus. And number three, it is tympanum or eardrum. Tympanum or eardrum. So, the three parts of the external ear are ear pinna or auricle. Number two, external auditory meatus. And number three, it is tympanum or eardrum. Next, about middle ear. This middle ear is a small air-filled cavity present inside a bone called as temporal bone. The middle ear is a small air-filled cavity present inside a skull bone called as temporal bone. Now, in this ear cavity, middle ear cavity, we find a chain of three bones called as ear ossicles. A chain of three small bones present inside the middle ear cavity, you call them as ear ossicles. Presence of three ear ossicles is a mammalian characteristic because all other gnathostomes possess only a single ear ossicles, in particular amphibians, reptiles and aves possess a single ear ossicle, whereas mammals possess three ear ossicles. Then when you go back to fishes where middle ear is absent, so there is no question of ear ossicles at all. Now, what are those three ear ossicles? From inside to outside, the three ear ossicles are malleus, incus and stapes. The three ear ossicles from inside to outside are malleus, incus and stapes arranged in a chain like fashion. Now, look at the stapes. Stapes is the smallest bone in the body. Now, to remember very easily the order, remember it as Miss. Next, it is about inner ear. Inner ear is otherwise called as labyrinth. Labyrinth. Now, this inner ear in turn has three parts. What are they? Number one, it is cochlea. Number two, it is semicircular canals. And number three, it is vestibule. So, the three parts of the inner ear are cochlea, semicircular canals and vestibule. So, this is outline details of the structure of the ear. So, external ear with ear pinna, external auditory meatus and tympanum or eardrum. Middle ear has a chain of 3 ear ossicles enclosed in the cavity of one temporal bone of the skull. Then inner ear, otherwise called as labyrinth, has 3 parts, namely cochlea, semicircular canals and vestibule. Let us learn about the structure of external ear. As we already discussed, external ear has 3 parts, that is, number 1, it is ear pinna, a lobe like structure specialized in receiving sound vibrations from the air. Now, what is it made of? It is made of cartilage, elastic cartilage, and is covered by skin, specialized in receiving. Now, it is made of elastic cartilage, a flap of elastic cartilage, and it is covered by skin. Next, the second part of it is nothing but external auditory meatus. It is nothing but a curved tube like structure. Number 2, external auditory meatus, a curved tubular structure. This auditory tube or auditory canal ends up in a delicate membrane structure, nothing but tympanum or eardrum. The third part of it, eardrum, otherwise called as tympanum or tympanic membrane. This is a semi-transparent membrane made of connective tissue. Externally, it is covered by skin and internally covered by a mucous membrane. These are the 3 parts of external ear. Earlobe, then curved tube, external auditory meatus, and followed by tympanum. Next, it is about the middle ear. Middle ear is small air filled cavity, we discussed it already, and it is lodged or placed inside one temporal bone of the skull. Now, in this middle ear cavity, we find a chain of 3 ear ossicles. So, what are those 3 ear ossicles? We learned the names already. Number 1, it is malleus, and number 2, it is incus, and number 3, it is stapes. Malleus, incus and stapes. These are all nothing but modified skull bones. When you talk about embryonic origin of malleus, it is derived from one articular cartilage, then incus is derived from one quadrate, and stapes is derived from a bone called as hyomandibular. So, what the embryonic origin of the 3 ear ossicles? Malleus is a modified bone called as articular, incus is a modified bone called as quadrate, and stapes is a modified bone called as hyomandibular. So, this is about mammals. In particular, we are talking about human beings. When we go back to amphibians, reptiles and aves, the only ear ossicle present is columella oris. That columella oris is nothing but modified hyomandibular bone. That means the stapes is comparable to columella oris based on its embryonic origin. Next, of course, also function. This is also playing a role in transmission of sound vibrations from the external ear, that is from the tympanum to the inner ear. Now, based on the shape, malleus is called as hammer, incus is called as anvil, and stapes is called as stirrup. So, hammer is, malleus is hammer, incus is called as anvil, and stapes is called as stirrup. To remember easily, malleus, incus, stapes, I asked you to remember it as miss, and now this is has, hammer, anvil, stapes. Miss, has, the order, very easy to remember. Okay. Then, what is the significance of these ear ossicles, the chain of the 3 bones? To convey the sound vibrations received by the tympanum to the inner ear. Transmission of sound vibrations from the tympanum to the inner ear is the function of the ear ossicles. So, that is about, and the next thing is about middle ear cavity. This borders of middle ear cavity. This middle ear is separated from the external ear by tympanum or eardrum, and also separated from the inner ear by a bony partition with 2 membrane covered openings. The 2 membrane covered openings are one oval window, otherwise called as fenestra ovalis, and next, and the other one is fenestra rotundus or round window. So, the 2 partitions or boundaries of middle ear cavity are, one is tympanum that separates middle ear cavity from the external ear, and the other one is a bony partition with 2 membrane covered openings, that is oval window or fenestra ovalis, and the other one is round window, otherwise called as fenestra rotundus. And the next thing is about middle ear cavity. Now, the middle ear cavity is connected to the pharynx, or it opens into the pharynx. Which part of the pharynx? You studied that the pharyngeal cavity has 3 parts. They are nasopharynx, located above the soft palate. Next, oropharynx, located just below the nasopharynx, that is below the soft palate, and laryngopharynx. Now, this middle ear cavity opens into the nasopharynx, nothing but part of the pharyngeal cavity located above the soft palate. Okay. Then, what is the significance of this? Means, it is to maintain the integrity of tympanum, protection of tympanum, otherwise we say. We already discussed that tympanum is a very delicate membrane and it is receiving sound vibrations from outside. So, there are chances of rupture of this tympanum in case of high vibrations. Now, because of this opening of the middle ear cavity into the nasopharynx, the air also enters into the middle ear cavity, thereby applying pressure on the tympanum from inside. That means, there is an equal pressure on both the sides of the tympanum that helps in maintaining the structure of the tympanum, equalizing the pressure and protection of the tympanum is the major significance of opening of middle ear cavity into the nasopharynx through the eustachian opening. Completed learning about external ear and middle ear. Now, let us also learn about the structure of inner ear or labyrinth. This inner ear or labyrinth is a bony structure made of bone basically. So, you call it as bony labyrinth. This in turn is divided into number of compartments by connective tissue partitions or membrane partitions, and all the membrane partitions present inside the bony labyrinth together you call it as membrane labyrinth. Inner ear structure, let us call it as bony labyrinth, an outer bony structure, and with membranes inside, let us call it as membrane labyrinth. Now, the cavities of this labyrinth or inner ear are filled with a colorless fluid. You call it as a lymph. It could be either endolymph or perilymph. Now, so this inner ear in turn has 3 parts, as we learned the names already. The very first thing we discuss here is about cochlea. What is this cochlea? Look at the word, look at the labeling. Cochlea is nothing but a watch spring like structure. It is a watch spring like coiled tubular structure. Though we call the bony part of this cochlea as a single tube, it is said to be actually a tube within 3 tubes. Inside this cochlea, there are 3 tubes due to 2 membrane partitions. So, what are those 3 tubes present inside this cochlea? So, we are seeing that cochlea is a watch spring like coiled tube, but it has 3 tubes in fact inside it. What are those 3 tubes? They are, number 1, it is scala vestibuli, and number 2, it is scala media, and number 3, it is scala tympani. So, the 3 tubes present inside the cochlea are scala vestibuli, scala media, and scala tympani. Now, these 3 tubes are separated from one another by 2 membrane partitions that constitute the membrane labyrinth of cochlea. So, what are those 2 membrane labyrinths? To begin with, the scala vestibuli is separated from scala media by a membrane called as Reissner's membrane. The same way, scala media is separated from the lower scala tympani by another membrane partition called as Basilar's membrane. Now, out of these 3 tubes, vestibuli and tympani are filled with a fluid called as perilymph, whereas the fluid present in the media, you call it as endolymph. The next thing is about scala vestibuli and media. The scala vestibuli ends up in the oval window or fenestra ovalis. The same way, tympani ends up with round window or fenestra rotundus. The next thing about Basilar's membrane, which is separating media and tympani. On the Basilar's membrane, there is a sensory ridge called as organ of Kārti. Present on the Basilar's membrane, there is a sensory ridge called as organ of Kārti. What is it made of and what is the significance of it? This organ of Kārti is made of several layers of hairy cells, made of several layers of sensory hairy cells arranged one above the other. Now, these sensory hairy cells are also provided with stereocilia on their free surfaces. Now, on the whole, this organ of Kārti, nothing but sensory ridge, is covered by an elastic membrane called as tectorial membrane. This organ of Kārti is specialized in perceiving or receiving the sound vibrations. This is about cochlea. Then what next? Let us learn about the semicircular canals. Presence of 3 semicircular canals at angles to each other is a characteristic feature of all the gnathostomes. Semicircular canals. 3 in number, arranged at angles to each other. Now, the base of each of the semicircular canal is swollen. Now, the swollen base of the semicircular canal is called as ampulla. The swollen base of each semicircular canal is called as ampulla. Now, inside each ampulla, there is also a sensory ridge called as crista. So, when there are 3 semicircular canals, how many ampullae are there? That means there are 3 ampullae. Inside each ampulla, there is one sensory ridge called as crista. Crista singular, cristae plural. You also call it as cristae ampullaris because they are present inside the ampullae. Then, what is the function of this cristae ampullaris? They are sensitive to rotation. The cristae, the hairy ridges, sensory ridges present inside the ampullae are sensitive to, and they provide a sense of angular acceleration. Provide a sense of angular acceleration. So, that means these sensory ridges, that is cristae, are sensitive to like turning of the body, turning of your head side to side, and they provide a sense of angular acceleration. That is about semicircular canals. What next? It is nothing but vestibule, an oval shaped structure present between the semicircular canals and the cochlea. The third part of the inner ear. Vestibule. Now, vestibule is a bony labyrinth. Now, inside this vestibule, there are 2 membrane labyrinths. The 2 membrane labyrinths present inside the vestibule are called as saccule and utricle. Saccule and utricle are the 2 membrane labyrinths present inside the vestibule. Now, each of this membrane labyrinth of vestibule, that is saccule and utricle, also have one sensory ridge each. Now, the sensory ridge present inside this saccule and utricle are called as maculae. Each one has its own sensory ridge. So, maculae plural. Now, what is the function of this maculae? Maculae are sensitive to gravitation. They are sensitive to gravitation and they provide a sense of linear acceleration. Linear acceleration means it could be either vertical acceleration or horizontal acceleration. Now, about the 2 maculae. The macula of saccule provides a sense of linear acceleration, whereas the macula of utricle provides a sense of horizontal acceleration. The membrane labyrinths of vestibule, that is saccule and utricle, with their respective maculae, together we call it as otolith organ. So, otolith organ is sensitive to gravitation, where the macula of saccule provides a sense of vertical acceleration, whereas macule of utricle provide a sense of horizontal acceleration. Now, this otolith organ along with semicircular canals together is called as vestibular apparatus. Now, this vestibular apparatus is sensitive to body balance. Body balance. So, the otolith organ, that is the membrane labyrinth of vestibule plus semicircular canals. Together, you call it as vestibular apparatus. So, that controls body balance. So, about the sensory ridges, the organ of 𝗳𝗃, sensory ridge present on the basilar membranes of cochlea, is sensitive to sound vibrations. That means it is responsible for hearing. Whereas, the sensory ridges present in the semicircular canals, that is ampullae, the sensory ridges present in the vestibule, that is maculae, together responsible for body balance, that is staticity. Then, about supply of cranial nerves. When we look at this organ of 𝗳𝗃, the sensory fibers of cochlear branch of auditory nerve, you studied already about cranial nerves, the 12 pairs, where 8th pair of cranial nerves are called as auditory nerves or statoacoustic nerves. You know, they are sensory nerves, and each auditory nerve has two branches. One is cochlear branch and the other one is vestibular branch. So, the cochlear branch collects its sensory fibers from the cochlea, that is organ of 𝗳𝗃 of cochlea. The same way, the vestibular branch of auditory nerve collects sensory fibers from the vestibular apparatus. So, cochlear branch and vestibular branch together, it is auditory nerve supplying sensory impulses to the medulla oblongata. That is, in fact, at the junction between pons varolii and medulla oblongata. So, with this, the structure of the inner ear is completed. Let us learn a bit also about mechanism of hearing. So, when we studied about the structure of the ear, we learned that the ear pinna or auricle is specialized in receiving or collecting sound vibrations from the air. Now, these sound vibrations collected by the ear pinna travel through the external auditory canal that ends up in tympanum. Now, these sound vibrations generate vibrations on the tympanic membrane. Now, the vibrations generated on the tympanic membrane are received by the chain of ear ossicles, that is, the travel along malleus, incus and stapes. Now, from here, the sound vibrations are received by the fenestra ovalis, a membrane covered opening, where from these sound vibrations enter into the inner ear. As we already studied about, cochlea with its sensory ridge organ of 𝗳𝗃 is specialized in hearing. Now, as the vibrations from the fenestra ovalis enter into the cochlea, there are ripples created, that is, otherwise there are waves created in the perilymph and endolymph. Now, you know that vestibuli and tympani are filled with perilymph, whereas media is filled with endolymph. Now, the vibrations generated in the perilymph and endolymph apply pressure on the basilar membrane. Now, this creates ripples on the basilar membrane. Now, the hairy cells are pressed against the so-called tectorial membrane, which is covering the organ of 𝗳𝗃. Now, this tectorial membrane is elastic membrane that can be stretched, right? Now, the stretching of this tectorial membrane generates action potential in the hairy cells, and now the action potentials are now received by the cochlear branch of auditory nerve. Now, these action potential or nerve impulse will be received by medulla oblongata through auditory nerves, and now where information is processed and analyzed, accordingly it is hearing. Nose is specialized in two functions actually. One is respiration and the other one is olfaction, that is sense of. As we are learning about sense organs, let us learn about only the olfactory function of the nose. You already studied about the structure of the nose, that is nasal cavities, right? Each nasal cavity has three different regions you studied from base to the top. The lowermost region you studied as vestibular region, and followed by you studied it as respiratory region, and then you studied it as olfactory region. Each nasal cavity has three different regions, where the lowermost region, in fact surrounded by the external nostrils, you studied it as vestibular region. Then followed by the largest part, respiratory region. Then uppermost part of each nasal cavity you call it as olfactory region. Now this olfactory region is lined by specialized sensory epithelium, you call it as olfactory epithelium or Schneiderian membrane. Now this olfactory epithelium or Schneiderian membrane is made of pseudo-stratified epithelium. Olfactory epithelium or Schneiderian membrane lining the olfactory region of nasal cavity is made of pseudo-stratified epithelium. Now this epithelium in fact is made of three different types of cells. What are they? Number one, olfactory sensory cells, and number two, sustencular cells, otherwise called as supporting cells, and number three, basal cells. So the three types of cells in the olfactory epithelium are number one, basal cells, number two, olfactory cells, that is olfactory sensory cells, they are nothing but sensory neurons, and then it is supporting cells, otherwise called as sustencular cells. Now you can look at this epithelium made of single layer of cells, but looking like as if it is made of 3 to 4 layers of cells. That is why you are calling it as pseudo-stratified epithelium. Now let us discuss only about olfactory cells here. The cell bodies of olfactory cells, let us say these are the olfactory cells present in between the sustencular cells or supporting cells. Each olfactory sensory cell is a bipolar sensory neuron. Bipolar sensory neuron. What do you mean by bipolar sensory neuron? A bipolar neuron is a one that has one dendrite and one axon. One dendrite and one axon. Now the dendrite of each of the olfactory sensory cell is divided into 6 to 8 branches. You call them as sensory heads or olfactory heads. You know that dendrites are afferent fibers. They collect sensory impulses. Okay, and now these sensory impulses are now received by the cell bodies of olfactory cells. Now from the cell bodies of olfactory cells, they are collected by non-myelinated axons of these sensory cells. Now all these non-myelinated axons join together to form olfactory nerve, the first cranial nerve, and they convey sensory impulses to the cerebral hemispheres through olfactory lobes. So this is about sensory function or olfactory structure and function of the nose. So, a bipolar neuron is characterized by presence of one dendrite and one axon. Of course, axon is always 1. So, such neurons with one axon and one dendrite are called as bipolar neurons, and here olfactory sensory neurons belong to the same category. It is about the tongue, the other sensory structure specialized in sense of taste. You call it as gustation. You already studied about structure of tongue in detail under digestive system. You know that it is a muscular motile structure present on the floor of the buccal cavity, attached to the floor of the buccal cavity by a connective tissue strand. You call it as frenulum. Then when you come to the sensory structure function of this tongue, you studied about taste papillae, right? So, you studied about presence of 3 different types of taste papillae on the tongue. What are those 3 types of papillae? You studied as fungiform papillae present at the tip of the tongue and margins of the tongue. Then you also studied about filiform papillae present on the surface of the tongue, and you also studied about the third important type of papillae, that is circumvallate papillae present at the base of the tongue. They are nothing but sac-like structures present on the surface of the tongue, giving a rough appearance to the tongue. Now, present inside these taste papillae, there are taste buds. Present inside these taste papillae, there are taste buds. Taste papillae are sac-like structures present on different parts of the tongue. Now, inside these taste papillae, there are taste buds, but the taste buds are not present on all the papillae. So, when you look at the number of taste buds on the human tongue, generally there are 10,000 of taste buds. A few taste buds are also present on soft palate, pharynx, and also epiglottis. Now, when you look at the structure of a taste bud, it is an oval shaped structure. Each taste bud is an oval shaped structure, and it is made of 2 different types of cells. So, what are those cells? Number 1, supporting cells, and number 2, it is taste cells, that is sensory cells. Now, let us look at the structure of a taste bud. An oval shaped structure. The basic capsule of the taste bud is made of epithelial cells, modified epithelial cells. You are calling these epithelial cells as actually supporting cells, supported by supporting cells. Let us say this is supporting cell and these are sensory cells. Sensory cells specialized in perceiving sense of taste, which is nothing but a chemical stimulus. So, definitely they have dendrites specialized in receiving that chemical stimuli, and these dendrites are hair-like structures called as gustatory hairs. And now these hairs come out of this taste bud through an opening present on the top of this taste bud called as taste pore. So, this is pore, and through which the gustatory hairs, nothing but the dendrites of the gustatory sensory cells, come out and specialized in perceiving chemical stimuli. Now, the axons of these sensory cells once again join together. Let us learn about the structure and functional details of the skin, one of the 5 senses. We already learnt about 4 other special sense organs, that is eyes, ears, nose and tongue, and we are left with the skin. When you come to the study of skin, you call it as dermatology. Study of the structure and functions of the skin, we call it as dermatology. Then, before going to the structural details, let us have a glance at the overall functional aspects of the skin. You know that it is the outermost covering of the body. That means it is covering all the internal organs of the body. So, protection. Under tissues, you studied about adipose tissue associated with the skin, subcutaneous adipose. What for is it? Act as thermal insulator. Helps in homeothermy. Next, skin also plays a role in absorption, and skin plays a key role as a sensory structure above all these functions. Now, when you come to the structural details of the skin, human mammalian skin in particular, it has 2 different regions, where the outer region is called as epidermis, or you say outer layer is called as epidermis and the inner layer is called as dermis. Now, both the layers of the skin are different both structurally as well as functionally as well as by their embryonic origin. Okay. Now, let us begin with epidermis. Epidermis is derived from outermost extraembryonic membrane, that is ectoderm, whereas dermis is derived from the germ layer mesoderm. Now, about the details of the epidermis, we can learn the structural details of the epidermis here looking at the diagram itself. Now, in general, you find around 5 different layers in the epidermis, which is made of epithelium. Now, to begin with, the first and the basic layer of epidermis that rests on a basement membrane, you call it as stratum malpighi or stratum germinativum. Why do you call it as germinativum? Because it gives birth to the cells of this layer continuously undergo mitotic divisions and give rise to the upper layers of cells. Okay. So, you call this malpighi, otherwise as germinativum. Next, above this germinativum, you find around 6 to 7 layers of polyhedral cells together called as stratum spinosum. Why the name spinosum? Because different cells of this spinosum are interconnected with one another by pointed spine-like cell junctions. So, you call it as spinosum. Now, above this spinosum, you find a layer called as stratum granulosum. As the name itself indicating, it is provided with special granules that take basic strains called as keratohyalin granules. Keratohyalin granules present scattered in the cytoplasm, but not in the cell organelles. Okay. Now, the lipids present in these keratohyalin granules diffuse to the surrounding intercellular spaces, making it waterproof. Next, above this granulosum, you find another layer called as lucidum, where these keratohyalin granules are dissolved and make the cells very much transparent and waterproof. Next, above this stratum lucidum and the uppermost layer of very much flattened scale-like cells called as stratum corneum. The outermost layer of the cells which are very much flattened and scale-like, you call it as stratum corneum. Why the name corneum? Because the cytoplasm of these cells is mostly occupied by a dead and horny protein, you call it as keratin. Previously, we studied this stratum corneum as a layer of dead cells, but now people say that they are not dead cells, though there is no nucleus, but they are living cells and secrete specific hormones as well that control the rate of cell divisions by the stratum Malpighi. Okay. Overall, when you look at the function of the epidermis, it is specialized in protection. Next, in the same epidermis, in the lower layers of cells, you also find pigment melanin. Those cells of the epidermis that contain this dark colored pigment melanin, you call them as melanophores. And what for melanophores are? What for melanin is? Protection from UV rays. Okay. When mostly this corneum, lucidum and granulosum are specialized in mechanical protection of our body. Melanophores present in the same epidermis protect our body from the bad effects of UV radiations, that is mostly mutations. And next, below this epidermis we find one dermis. Now, this dermis is basically made of connective tissue and we can differentiate this dermis again into two regions, where the outer region of the dermis that lies close to or just below the epidermis as dermis papillosum, or you say papillary layer, and now the lower layer of the dermis you call it as reticular layer. So, the dermis, which is basically made of connective tissue, has two regions, an upper region that is close to the epidermis or present immediately below the epidermis, you call it as papillary layer, and the lower layer you call it as reticular layer. Now, let us see the difference between this papillary layer and reticular layer. As we discussed, papillary layer is close to the epidermis. Now, look at this papillary layer. This papillary layer is projecting into the epidermis in the form of papillae. In the form of papillae, hence you are calling it as papillary layer. Okay. Now, when you come to the structural details of this papillary layer, it is made of areolar connective tissue. You studied already areolar connective tissue. It is one of the loose connective tissue proper. That means it is highly vascular, provided with collagen, reticular as well as elastic fibers and different types of cells as well. Now, apart from that, what we have to discuss here is skin as a sensory organ. So, you can find different nerve endings of different nerve fibers that play a key role in perceiving the sense of pain. Then, you also find cup-like structures called as mucinous corpuscles, which are specialized in sense of touch. Okay. So, in this papillary layer, which is made of areolar connective tissue, you find free nerve endings that play a role in pain perception, that is receiving the sense of pain, and you also find sensory capsules called as mucinous capsules that play a key role in perceiving the sense of touch. The same way, when you come to this lower layer of dermis called as reticular layer, it is made of dense fibrous tissue. It is made of dense fibrous tissue. Dense fibrous tissue means what? The matrix is completely packed with bundles of collagen fibers. Apart from that, there are also elastic fibers scattered in different directions. Okay. Now, these collagen fibers gives mechanical strength to the skin. Whereas, what for elastic fibers are? Elastic fibers help in regaining the shape of the skin. So, because of the collagen fibers, skin can be stretched to a certain extent, and because of the elastic fibers present there, the skin can be back to its shape. But with increasing age, due to continued stretching of these collagen fibers, you find wrinkles in the skin of old aged people, because they gradually, that is collagen fibers, gradually lose their mechanical strength. Now, besides this, what else do you find in this reticular layer of dermis? Means you find sweat glands, you can see the coiled simple tubular gland, and you also find sebaceous glands, and you also find erector pili muscles associated with the hair follicles. You studied already about erector pili muscles, once again in the smooth muscle category. Yes, they are the smooth muscles associated with the hair follicles and responsible for erection of the hair you studied, right? And next, you also find adipose cells. Okay, besides this structural details about sensory function. In this reticular layer of the dermis, there are sensory structures, cup-like sensory structures. You call them as Pacinian corpuscles, and these Pacinian corpuscles are specialized in pressure perception. So, overall, when you look at at a glance, when we discuss about few receptors present in the dermis, you find mucinous corpuscles specialized in sense of touch in the papillary layer. In the same papillary layer, you find nerve fibers, that is free endings of the nerve fibers that are specialized in pain perception. The same way, in the reticular layer of the dermis, you also find sensory structures called as Pacinian corpuscles, which are specialized in pressure perception. So, this is about the structure and functional details of the skin.